Let's start with a simple but critical question. What if a tiny, almost invisible impurity could shut down an entire pharmaceutical production line? Today, we're talking about nitrosamine contamination, a topic that has become a major concern across the pharmaceutical industry. In this video, I'll walk you through 5 common sources of nitrosamine contamination, and more importantly, the critical controls you must have in place to protect product quality and patient safety. So first, what exactly are nitrosamines? Nitrosamines are highly potent genotoxic impurities, meaning they have the potential to damage DNA and cause cancer. They form when amines react with nitrosating agents, such as nitrites. What makes them especially challenging is that they don't just appear during synthesis. They can form during manufacturing, storage, or even packaging. That's why regulatory agencies enforce extremely strict limits, often in parts per billion, due to their association with serious health risks like liver and gastric cancers. Let's begin at the source, your raw materials. Even when materials meet specifications, they can still carry trace levels of amines or nitrites. For example, solvents like DMF or NDMA may contain residual amines, while reagents or excipients might introduce nitrite impurities. Individually, these seem insignificant, but together, they can trigger nitrosamine formation later in the process. So the key here is control. Strengthen your supplier qualification. Perform targeted testing using techniques like GCMS or LCMS, and always request a formal risk assessment from vendors. Now let's talk about something widely practiced, solvent recycling. While it improves sustainability and reduces cost, it also introduces risk. With each reuse cycle, impurities like amines and nitrites can accumulate. And if your purification process, like distillation, isn't efficient enough, these precursors remain in the system. Over time, this creates the perfect environment for nitrosamine formation. To control this, limit the number of reuse cycles, implement additional purification methods like activated carbon, and most importantly, perform routine monitoring using mass spectrometry. Sometimes, the issue isn't what you use, but how your process is designed. Certain conditions significantly accelerate nitrosamine formation. These include acidic pH, high temperatures, and extended reaction times. If amines and nitrites are present under these conditions, the reaction becomes much more likely. That's why it's critical to avoid combining these components under acidic environments and to thoroughly evaluate your process. Using tools like FMEA, failure mode and effects analysis, can help map out exactly where these risks exist. Now here's a source that's often underestimated: water systems. Water can introduce nitrosating agents in 2 main ways. First, microbial activity in storage tanks can generate nitrites. Second, chloramines used in disinfection can degrade into reactive species like nitrous acid. Since water is used throughout manufacturing, even small contamination can have widespread impact. To manage this, maintain proper thermal sanitization, typically above 80 degrees Celsius, and regularly monitor nitrite levels in your system. Even after manufacturing is complete, the risk doesn't disappear. Nitrosamines can form during storage. This can happen due to interactions with packaging materials, especially if they contain trace amines in polymers or adhesives. Environmental factors like heat and humidity can further accelerate these reactions over time. That's why it's essential to conduct ICH stability studies, choose inert, low-leaching packaging materials, and strictly control validated storage conditions. Now let's connect everything we've discussed. Nitrosamine formation always comes down to 3 key elements: amines, nitrosating agents, and favorable conditions. If all 3 are present at any stage, Whether in raw materials, process, or storage, you have a risk. This is why control isn't just about one step. It requires a full life cycle approach, from sourcing materials all the way to the product sitting on the shelf. So how do you practically manage this risk? Start by asking simple but powerful questions. Are amines or nitrites present? Are the conditions suitable for reaction? Then move to monitoring. Implement validated analytical methods like LC-MS, MS to detect even trace levels. And finally, document everything: risk assessments, supplier data, and control strategies. Because in regulated environments, if it's not documented, it doesn't exist. To wrap up, nitrosamine contamination is not just a testing issue, it's a process understanding challenge. The goal isn't just to detect them after they form. The goal is to design processes where they never form at all. Because ultimately, every control you implement is about one thing: ensuring patient safety.